I was more in the semiconductor kind of area, but when I go to PhD, I started really the first time more really like in contact with biology. I started studying biology. I think it's really interesting thing to study, right? And I start from biology and I start go to cancer biology a little bit, like to see how can engineering help in to understand, develop new tools or develop the methods to help understand the cancer better. One of the biggest challenges in cancer is it's very complicated disease, which means it's very difficult to be defined by one or few molecules of the system, and the signatures of the disease might be changing from patient to patient. It's very complicated, right? That's why I think to establish this sort of multiplex or high throughput methods, essentially, it is one of the important way to discover cancer better or understand cancer better is to be able to know how the cancer cell in this complicated microenvironment, potentially everybody have different microenvironment parameters, and how do we have an efficient way to learn what the microenvironment important parameter is it, and then make a prediction on how the cancer cell will respond to, for example, therapeutic treatments or progressions of the cancer, like how does it all interplay. So since it's just a lot of information there, so I think one of the efficient way to really understand is just to build a workflow to be able to extract multiple information at the same time and use it against cancer. The focus we do for the PSOC is to study the complicated tumor microenvironments, study how the microenvironments and the cancer cell interplay and the influence with each other's function, behavior, stuff like that. Since it's very complicated environments, there's so many variables, factors within the microenvironment could influence the behavior of cancer cells. So we are establishing a lot of new techniques and assays to extract informations in this kind of complex environments to hopefully it can guide us how to build a better biomarker models and build a better cancer model system to study cancer.